Welcome to our deep dive, everyone, where we're going to be exploring the fascinating world of microbial control.
It's such an important topic.
Yeah, it really is. And, it's something that affects all of us in our daily lives.
Absolutely.
But, we might not always think about the science behind it.
Right. It's one of those things that's kind of hidden in plain sight.
Exactly. So today we're going to shine a light on all the different ways that we can control these tiny but mighty microbes.
And you might be surprised by the range of methods we use and just how much it impacts your everyday life.
Yeah, for sure. And I know I've learned a lot just in preparing for this episode.
Me too, actually. It's such a dynamic field.
Yeah, so let's jump right in. We've all heard those terms like sterilization, disinfection, antisepsis, but what do they actually mean?
Well, they all refer to different levels of microbial control.
Okay, so it's not just a matter of, cleaning something.
Exactly.
Okay.
Sterilization is the most extreme level we're talking about, completely wiping out all microbial life. Even those resilient bacterial spores. So you think surgical instruments, they need to be absolutely sterile.
Right, yeah.
Disinfection, on the other hand, focuses on reducing the number of pathogens, but it might not eliminate everything.
So like when you're wiping down your kitchen counter with a disinfectant?
Exactly. And then there's antisepsis, which uses antimicrobial chemicals that are safe for living tissue.
Oh, okay, so like hand sanitizer.
Exactly. Your trusty hand sanitizer.
Okay, so different situations call for different levels of microbial control.
Absolutely.
But why is all this so important? What's the big deal about a few microbes here and there?
Well, those few microbes can multiply quickly.
Oh, yeah?
And some can be quite harmful. Think about it. You touch a doorknob, then rub your eye, and suddenly you've introduced microbes from who knows where into your body.
Right, those everyday objects we touch, like doorknobs, phones, even like the pen I borrowed from you earlier.
Yep. Those are called fomites.
Fomites.
And they play a surprisingly significant role in disease transmission. Oh, yeah. You'd be amazed how long some viruses can survive on surfaces just waiting for an opportunity to jump to a new host.
All right, that's officially making me rethink my hand sanitizer habits.
Good.
So how do we decide what level of microbial control is needed in a given situation?
It all depends on the risk involved. For example, labs working with infectious agents use a system called biological safety levels.
Biological safety level.
BSLs for short.
Okay.
It's like a danger rating for germs.
Oh, interesting.
BSL-1 is for working with relatively harmless microbes,
Okay.
while BSL-4, the highest level, is reserved for highly dangerous pathogens like Ebola.
Wow. So that's pretty serious.
Absolutely. And as the risk increases, so do the precautions.
That's good to know that there are those protocols in place.
Yeah.
So once we've figured out, the level of control that we need, how do we actually, eliminate or at least control those microbes?
Well, we use two main approaches, broadly speaking, physical methods and chemical methods.
Okay, so let's start with the physical methods. What kind of strategies are we talking about here?
Think of it as harnessing the power of physics to combat microbes.
Okay.
One of the most common and effective methods is heat.
Heat, like boiling water to sterilize baby bottles.
Exactly. We can apply dry heat like in an oven,
Okay.
or moist heat, which is even more effective because it penetrates cells more readily.
Oh, okay.
Think about autoclaving, which uses pressurized steam to sterilize medical equipment.
Wow.
And of course, there's pasteurization, which uses heat to eliminate pathogens in milk and other beverages.
So are we basically like cooking those little buggers?
In a way, yes.
What?
We're disrupting their cellular structures and essentially rendering them inactive.
That's fascinating. What about other physical methods?
We can also use extreme cold. Refrigeration slows down the growth of most microbes, while freezing can completely stop growth and even kill some organisms.
So my freezers is like a microbial time machine,
That's a great way to put it.
pausing their growth indefinitely.
Ultra-low temperature freezers are used to preserve lab specimens and other sensitive materials, essentially putting those microbes on hold.
That's so cool. What other physical tools do we have in our microbial control arsenal?
We can use high-pressure processing,
High pressure processing.
which is becoming increasingly popular in the food industry. It's a way to kill microbes without heat preserving the flavor and nutrients in food.
Oh, interesting.
And then there's desiccation, basically drying things out to make it inhospitable for microbes.
Like beef jerky.
Exactly.
Hmm?
Adding salt or sugar also helps with desiccation.
Okay.
They draw water out of the cells, making it difficult for microbes to survive. That's why jams and jellies last so long.
Interesting. And what about those freeze-dried astronaut ice cream packs?
That's a great example of lyophilization, which combines freezing and drying for long-term preservation of food and lab materials.
Wow, that's amazing.
Yeah.
Are there any other physical methods that we should know about?
One more important, radiation.
Radiation.
We use ionizing radiation like gamma rays to sterilize heat-sensitive materials.
Okay.
Like medical devices and even some foods. It's a powerful and highly effective method.
Radiation sounds a bit intense.
Yeah.
What about non-ionizing radiation, like UV light?
UV light is excellent for surface sterilization.
Okay.
You've probably seen those UV lamps used in hospitals to disinfect rooms.
Right.
It can't penetrate surfaces, but it's very effective at killing microbes on exposed areas.
So we've got all these physical methods, from heat and cold to radiation and drying.
Right.
What about the chemical methods? Are we talking about like bleach and hand sanitizer here?
Those are certainly part of it.
Okay.
But the world of chemical microbial control is vast and varied. Let's start with some of the classics, like heavy metal.
Heavy metal, like the music.
Not quite. We're talking about metals like silver, copper, and zinc, which have been used for centuries to disinfect and preserve things. Think about those silver-infused bandages you see at the pharmacy.
Oh, right. Silver has those natural antimicrobial properties, but aren't some heavy metals toxic?
You're right. Some can be like mercury. So we have to be careful about their use. But in controlled settings, they can be very effective.
Makes sense.
Yeah.
What other chemical weapons do we have in this microbial battle?
We have halogens like chlorine and iodine. They're incredibly effective disinfectants. Bleach, for example, is a chlorine compound.
Right.
And iodine is used as an antiseptic.
Ah, yes. The familiar sting of iodine.
Exactly.
A badge of honor from childhood scrapes.
Then there are alcohols like ethyl alcohol and isopropyl alcohol.
Okay.
They're the main ingredients in hand sanitizers and work by denaturing proteins and disrupting cell membranes.
So rubbing alcohol is basically dissolving those little buggers.
You could say that. And let's not forget phenolics,
Phenomenal.
which are commonly found in household cleaners, mouthwashes, and even some crop preservatives.
Okay, those sound a bit more intense.
They're powerful disinfectants, but some, like triclosan, have become controversial due to concerns about their potential impact on human health and the environment.
Right. It's a good reminder that, even with microbial control, there can be trade-offs. Speaking of which, are all chemical methods created equal?
Not at all. Each chemical has its own strengths and weaknesses, and effectiveness can depend on factors like concentration exposure time and even the type of microbe we're targeting.
So choosing the right method is key.
Absolutely.
What other chemical options do we have?
Well, there are surfactants, which are basically soaps and detergents.
Okay.
So yes, even washing your hands is a chemical method of control.
Oh, interesting.
They work by lowering the surface tension of water, allowing it to penetrate and lift away dirt and microbes.
So it's more about the scrubbing action than the soap itself.
Exactly. But some soaps also have antimicrobial properties, especially those containing antibacterial agents. Then there are quaternary ammonium compounds, or QUATs for short,
Quats.
which are commonly found in cleaners and oral rinses. They disrupt cell membranes.
Okay, I think I've heard of those.
You probably use them every day without even realizing it. They're in many household cleaners and disinfectants.
Wow, so many chemical options. What about those really tough microbes like bacterial spores? Do we have anything that can handle those?
For serious sterilization, we use alkylating agents like glutaraldehyde and formaldehyde.
The formaldehyde, isn't it used for embalming?
Yes, it is. It's a very effective sterilant, but it can also be quite toxic, so it's not something you'd use for everyday cleaning.
Right. That makes sense. Are there any eco-friendly options for chemical control?
Absolutely. Peroxygens like hydrogen peroxide are very effective and break down into harmless byproducts like water and oxygen.
So hydrogen peroxide is like a green warrior in the fight against germs.
Exactly. And then there's pressurized carbon dioxide, which is gaining popularity as a sterilant for delicate medical devices and even transplanted tissues.
Wow.
It's non-toxic, non-flammable, and very effective.
It sounds like we've got a whole arsenal of chemical weapons at our disposal.
We do.
But with so many options, it must be tricky to know which one to choose.
That's where things get really interesting. The choice of method depends on several factors, like the type of microbe, the level of contamination, and even the environmental conditions. It's not just about grabbing the strongest disinfectant off the shelf.
So we've covered a lot of ground already: physical methods, chemical methods. It's clear that there's a lot more to microbial control than meets the eye.
Absolutely. And we've only just scratched the surface. There's so much more to explore, including how we test the effectiveness of these methods and the challenges we face in a world with increasing antibiotic resistance.
That's a great point. And we'll definitely delve into those topics in our next deep dive.
Looking forward to it. Welcome back to our deep dive into microbial control.
Before the break, we were talking about all the different ways that we can control microbes.
It's amazing how much science goes into something we often take for granted.
It really is. But before we move on, I have to ask, how do we actually know what level of microbial control we need in different situations? Is there like a cleanliness standard that we aim for?
That's a great question. The level of control really depends on the context.
Okay.
For example, a scalpel used in surgery needs to be absolutely sterile,
Right.
while a doorknob in your home doesn't require the same level of stringency.
Yeah, that makes sense.
Yeah.
So how do we determine these different levels of cleanliness, especially in places like hospitals where the stakes are so high?
Well, in healthcare settings, medical items are actually categorized based on their risk level for transmitting infections.
Okay.
We have critical items, semi-critical items, and
Yeah.
non-critical items.
Okay, let's unpack that a bit.
Yeah.
What exactly are critical items?
These are the things that come into direct contact with sterile tissues or the bloodstream.
Okay.
So think surgical instruments, implants, catheters. Because
Okay.
of the high risk of infection, these items must be sterilized.
So no room for error there.
Exactly.
Right.
What about semi-critical items?
Semi-critical items.
Those come into contact with mucous membranes or non-intact skin.
Okay.
Examples include endoscopes, respiratory therapy equipment, and certain dental instruments.
Oh.
For these, we aim for high-level disinfection. That means they need to be free of all vegetative bacteria, fungi, and most viruses.
Wow, that's pretty intense. And lastly, what falls under non-critical items?
Those are the things that only come into contact with intact skin.
Okay.
Think blood pressure, cuffs, stethoscopes, and bed rails. These items require a lower level of disinfection, usually with low-level disinfectants that target the most common bacteria and some viruses.
So it's like a hierarchy of cleanliness based on the risk of infection.
Exactly. And this system helps healthcare professionals make informed decisions about which microbial control methods are appropriate for each situation.
That makes a lot of sense. So
Yeah.
I've got our cleaning protocols in place, and we're using the right disinfectants for the job. But how do we actually know that those disinfectants are effective? We talked about the diffusion test earlier,
Right.
but is there more to it than that?
You're right. Testing is a crucial part of this whole process. We need to make sure those disinfectants are doing what they claim to do.
Yeah, otherwise we're just spraying chemicals around with no real guarantee of safety.
Exactly. One way we assess effectiveness is by determining the germicidal activity of a chemical.
Okay.
We classify germicides into 3 levels: high, intermediate, and low.
So it's another ranking system like those BSLs we talked about earlier.
Very similar. High-level germicides are the heavy hitters. They can kill vegetative cells, fungi, viruses, and even those tough bacterial endospores.
Wow.
Given enough exposure time, they can even achieve sterilization.
That's amazing. What about intermediate level germicides?
They're a step down. They can't kill all viruses and aren't as effective against endospores, but they can still knock out most common pathogens.
Okay, and low level germicides.
Those are your everyday disinfectants. They target vegetative bacteria and some enveloped viruses, but aren't effective against endospores.
Got it. So we've got these different levels of germicidal activity. But how do we actually test these chemicals to make sure they live up to their claims?
There are several standardized tests we use. One classic method is the phenol coefficient test. It compares the effectiveness of a disinfectant to that of phenol, a well-known antimicrobial agent.
So phenol is like the benchmark for disinfection.
It was historically used as the standard, yes. If a chemical has a phenol coefficient greater than 1, it means it's more effective than phenol at killing bacteria.
Okay,
However, this test has limitations and isn't always reliable.
I see. What other tests are there?
More widely used methods include the disk diffusion method, which we discussed earlier, and the use dilution test.
Okay.
The use dilution test measures how well a disinfectant works on a contaminated surface.
So the disk diffusion test is for screening different disinfectants, and the use dilution test is more about real-world applications.
Precisely. And then we have in-use tests, which are conducted in actual clinical settings to monitor the effectiveness of disinfectants in practice. These tests help ensure that protocols are being followed correctly and that the disinfectants are still effective over time.
That's so important. I can imagine that disinfectants can lose their potency if they're not stored correctly or if they're diluted too much.
You're absolutely right. Factors like concentration, temperature, and even the presence of organic matter can impact a disinfectant's effectiveness. That's why it's crucial to follow the manufacturer's instructions carefully and monitor their effectiveness regularly.
It really highlights the complexity of microbial control.
It does.
We've covered physical methods, chemical methods, levels of cleanliness, and now testing procedures. It's a multifaceted field.
It is, and it's essential to modern life, especially in healthcare. But it's not just about hospitals. Microbial control plays a crucial role in other areas like food preservation, which we'll discuss after a cue.
Yeah, I'm really interested to hear more about that.
It's a fascinating topic.
So, we've talked a lot about keeping things clean and sterile, especially in those healthcare settings. But what about the food we eat?
Right.
How does microbial control play a role in, keeping our pantries and refrigerators safe?
Well, food preservation is another fascinating area where microbial control is absolutely essential. I mean, after all, humans have been battling food spoilage for centuries long before we even knew what microbes were.
That's right. We've been drying, salting, pickling food for ages.
Exactly.
So I guess those were like some of the earliest forms of microbial control, even if we didn't really, realize it at the time.
Exactly. And those traditional methods are still used today alongside more modern techniques like refrigeration freezing and canning.
Canning always reminds me of my grandmother's homemade preserves.
Oh, nice.
There's something magical about capturing the flavors of summer in a jar.
And that magic is partly thanks to microbial control.
Oh.
The canning process involves heating food to a high enough temperature to kill any lurking microbes,
Right.
and then sealing it in an airtight container to prevent recontamination.
So it's like creating a little time capsule,
Right.
preserving the food and preventing any unwanted microbial guests from, crashing the party.
Exactly.
But I've also heard that canning can be kind of risky if it's not done properly.
That's true. If the food isn't heated thoroughly or the jars aren't sealed correctly, you can create an environment where harmful bacteria like Clostridium botulinum can thrive.
Right. Botulism is definitely not something you want to mess with.
No.
But what about other methods of food preservation? We talked about chemical preservatives earlier.
Right.
How do those fit into the picture?
Chemical preservatives are widely used in processed foods to extend their shelf life and prevent spoilage. We discussed some common ones like sorbic acid, benzoic acid sulfites, and nitrites.
Right. And we touched on the potential health concerns associated with some of those, particularly nitrites. Are there any regulations surrounding their use?
Absolutely. Government agencies like the FDA here in the United States set strict limits on the types and amounts of preservatives that can be added to food.
Okay, that's good to know. So it's not just a free-for-all when it comes to adding preservatives.
Right.
There are regulations in place to ensure safety. But, as consumers are increasingly interested in natural and less processed foods, are there any alternatives to these chemical preservatives?
There's a lot of interest in finding natural alternatives, and we did mention niacin and natamycin earlier. These are naturally occurring preservatives produced by bacteria and fungi, respectively.
Oh, wow.
Niacin is commonly used in cheese making, and natamycin helps prevent fungal growth in various foods.
So nature has its own arsenal of microbial control weapons.
It does.
That's pretty cool.
Yeah.
Are there any other exciting advancements in food preservation that you're particularly excited about?
There's a lot of research being done on innovative non-thermal processing methods.
Yep.
These techniques can kill microbes without relying on heat or chemical preservatives. Some examples include high-pressure processing and pulsed electric fields.
Wow, it sounds like the future of food preservation is quite high-tech.
It is.
But no matter the method, it all comes back to that fundamental principle controlling microbial growth.
You've hit the nail on the head. It's a constant dance between us and the microbial world.
Yeah.
We need to stay one step ahead to ensure our safety and well-being, whether it's in a hospital, our kitchens, or the food we consume.
This has been such a fascinating journey into the world of microbial control.
It has.
Before we wrap things up, are there any key takeaways you'd like our listeners to remember?
I think the most important takeaway is that microbial control isn't a one-size-fits-all approach.
Okay.
It's about understanding the specific situation, assessing the risks, and choosing the most effective and appropriate method from the toolbox we discussed today.
And perhaps a newfound appreciation for those everyday acts like washing our hands,
Absolutely.
which play such a critical role in keeping those microbes at bay.
Simple actions can have a huge impact when it comes to preventing the spread of infections.
Well said. So to our listeners, next time you reach for that hand sanitizer or admire the long shelf life of your favorite snack, take a moment to appreciate the intricate world of microbial control at work. It's a fascinating and constantly evolving field that touches every aspect of our lives.
And as we continue to explore and understand the microbial world, we're sure to discover even more innovative ways to manage and coexist with these microscopic forces.
It's an exciting time to be learning about microbial control. Thank you for joining us on this deep dive. We hope you found it insightful and engaging.
Until next time, stay curious and stay informed.